Hello everyone, welcome to the next episode of my science podcast. Today I'm delighted to welcome Professor Dame Nancy Rothwell, who is a British physiologist who is recognised for her research identifying mechanisms of energy balance regulation, obesity and cachexia, as well as her work on neuroscience, which has contributed towards major advances in the understanding and treatment of brain damage in stroke and head injury. She has been President and Vice-Chancellor of the University of Manchester since 2010, and she was elected Fellow of the Royal Society in June 2004. She was also made Dame Commander of the Order of the British Empire in June 2005 in recognition of her services to science. She is a member of the Physiological Society and a Fellow of the Academy of Medical Sciences. She was the founding President of the Royal Society of Biology and has also served as co-chair of the Prime Minister's Council for Science and Technology. And she has also served as a non-executive director of AstraZeneca. She is currently a Deputy Lieutenant for Greater Manchester, Chair of the Oxford Road Corridor Board and a member of the Greater Manchester Local Enterprise Partnership Board and the Northern Powerhouse Partnership Board. Most recently, since September 2020, she's been the chair of the Russell Group, which represents 24 of the leading universities in the UK. So it is a delight to welcome her today, and I really look forward to our meeting. Hello everyone, welcome to the next episode of my science podcast. So I've already done the introduction, but so we'll crack on straight into the questions. So to start off with, may you tell us about your early work on energy balance, such as the role for brown adipose tissue in diet-induced thermogenesis and resistance to obesity by the ventromedial hypothalamus?
Yeah, so in a nutshell, we did a lot of work rather than trying to understand why some people and some animals get fat, why some don't. In other words, why some people, and I happen to be one of them, seem to be able to eat whatever they want to and never put on weight. And what we found is that certainly animals and probably some people can burn off excess calories. And to do that, they don't have to exercise, their metabolism just goes up. And again, certainly in animals, there is no evidence for it in humans. That may be partly due to a tissue called brown fat, brown adipose tissue. And it's not at all like the normal white fat, which is where we store energy. Brown fat does the opposite, it burns off energy. It's got a really high metabolism, much higher metabolism than muscle. And we showed that in some cases, animals and probably people, when they eat more, they switch up their brown fat and burn it off and therefore stay thin. And probably brown fat's main function has been to keep babies warm because babies can't shiver. But also it's very important in hibernating animals because hibernating animals let their temperature drop down very, very low, sometimes below 10 degrees. When they wake up, they have to warm up really quickly. So they switch on their brown fat very, very quickly. And the point about the ventromedial hypothalamus, that's a part of the brain. It's not the only part involved in regulating body weight and appetite, but it is particularly important in driving feeding behaviours. That and the lateral hypothalamus, but obviously there's lots of brain areas that are involved. And one of the things we did show was that stimulation of the ventromedial hypothalamus can switch on brown fat and therefore potentially cause weight loss. And following that many years, people have been looking at ways of switching on brown fat as a dieting mechanism.
Fascinating. So we'll move on to your later work. So yep. You discovered that an inflammatory molecule called cytokine interleukin 1, or IL-1 for short, is made in the brain after a stroke or injury. And the natural blocker of this is called IL-1 receptor antagonist, or IL-1RA, which we'll probably refer to it as in the rest of this podcast. And so this not only increases stem cell proliferation, but also significantly enhances neuroblast migration and the number of newly formed neurons after cerebral ischemia. So this is where there's a restriction of blood supply to a region. So may you tell us about the clinical trial you ran for this receptor antagonist?
Yeah. So first of all, just to say that this inflammatory molecule, IL-1, we all have it. It's really important, and its main function is to kill off infections. And it's the key reason you feel pretty awful when you've had flu or a cold or something else. So it's a good thing. It's part of our defense. But we know that in a lot of diseases, they're due to overactivation or prolonged activation of our own defense mechanisms. Good example is arthritis, which is where our own body's defenses start to attack our own tissues. And a stroke wasn't thought to be one of those. And of course, a stroke is, as you say, where there's reduced blood supply to the brain. But we did find that there's increased production of IL-1, not just in the brain, but in the body as well to some extent, and that this naturally occurring blocker reduced the damage. Now, we don't know exactly how it reduces the damage. There's probably lots of reasons, some of the ones you suggested, but also we think that IL-1 acts on certain types of cells in the brain called glia, and the glia can release toxins that kill the nerve cells. And blocking IL-1 with the antagonist, and its medical name is Kineret, or anakinra, it's often called now, that stops that happening. But what we found in a number of studies that using this IL-1 receptor antagonist did seem to be beneficial. We've now carried out about 4 trials, 2 in stroke and 1 in subarachnoid hemorrhage, which is a bleeding in the brain. And subarachnoid hemorrhage actually has very similar consequences to a stroke, because with a stroke, the brain's starved of oxygen because there's a blockage in a blood vessel, normally a clot, whereas in a hemorrhage, of course, it's starved of oxygen because the blood vessel bursts and bleeds. And in both of those, blocking IL-1 seems to be helpful. These were all relatively small trials. Now, we were just starting a big trial in hemorrhage, COVID struck. And so, of course, we had to halt the trial. And I don't know if and when we're going to be able to restart it. And interestingly, the IL-1 receptor antagonist was moved to studies of COVID. And it looks as though it may be beneficial in COVID, because one of the things about COVID is a lot of people die, not particularly from the infection, but for an overactive immune response. Their immune response goes into overdrive. And so you may have heard terms like cytokine storm. This is when there's too much IL-1, too much IL-6 produced, and that again attacks our own bodies. And so the IL-1 antagonist has been tested and shown to be potentially beneficial in those. Now, we hope to go back to the trials for brain disease. There are also some trials that were started that got halted and will probably continue again in both dementia, because we think of dementia as being Alzheimer's, but actually a lot of dementia is called vascular dementia. It's caused by lots and lots of tiny, tiny little strokes that you might not even detect, but together they contribute to a gradual damage to the brain and a decline in function, although that's a chronic disease. And the other condition it's been looked at for is what's called birth asphyxia, because some babies, if the mothers are not healthy, are deprived of oxygen just before or during birth, often because of a failure of the placenta, and they get exactly the same things as happens in a stroke, not enough oxygen to the brain. So there's another trial in another part of the world going on into that. So we're hoping we're going to be able to go back to those trials.
Fascinating, and it's wonderful that it's so diverse and it could be used for so many different diseases.
Potentially.
Yeah, and you've mentioned Alzheimer's, well, you mentioned dementia, but for Alzheimer's, there's a greater therapeutic challenge here, as I read in one of your papers, as anti-inflammatory agents might reduce the probability of developing this disease or slow its progression, but prospective clinical trials of non-steroidal anti-inflammatory drugs or inhibitors have been disappointing. So why could this be?
Yeah, and we don't know exactly what that is. The early studies came from looking at people who were on chronic treatment with anti-inflammatory agents, steroids maybe, aspirins, paracetamol, and they have a lower incidence of Alzheimer's. And so it was suggested it might have been because of those anti-inflammatory agents. And then a prospective study is where, rather than looking backwards at what happened, you actually take a group of people and put them onto those treatments and see if they get Alzheimer's. And they didn't really show much difference. Now, what we don't know, and human studies, clinical studies are really complicated, lots of different factors. Does it mean that inflammation is not involved in Alzheimer's? There's a lot of evidence it is. Alzheimer's patients have higher levels of markers of inflammation, and if you scan their brains, they have evidence of inflammation, but it might be that. Were they the wrong anti-inflammatory agents? Because a lot of anti-inflammatory agents we've got have multiple effects. So, for example, aspirin has lots of different actions, not just anti-inflammatory. It affects clotting, for example. So it wasn't known, or possibly were they the wrong trials? Because one of the difficulties is translating from the laboratory to the clinical situation. Because in the clinical situation, you've got a huge diversity of different presentations, people of different ages, different stages. So the IL-1 antagonist is thought to be possible because it's very specific. All it does is block IL-1. It doesn't do anything else, and all the others do lots of things. The other thing that's complicating in inflammation is a lot of the drugs we use to target inflammation stop the production of molecules called prostaglandins. And prostaglandins, they're involved in pain, they're involved in inflammation. The reason you get your headache, why you take an aspirin or a paracetamol or something. But one branch of the prostaglandins is beneficial. And so by just stopping the whole lot, rather than just the bad branch or the good branch, you might negate the effects. And that's one of the difficulties in developing drug treatments. You need to ideally get something very specific. And this is where the IL-1 antagonist is particularly interesting because it's naturally occurring. And as far as we know, it doesn't do anything else apart from blocking the effects of IL-1, nothing else at all. So it might be more specific. Now, there is another issue, though, with the antagonist, in that it's quite a big protein and it's quite expensive. Aspirin's very cheap, whereas the antagonist, which is given chronically to people, particularly people with rheumatoid arthritis and children with inherited inflammatory conditions who are very, very ill and without this drug would probably die, and they're given it continuously. And it seems to be very safe, but it's not cheap. So that's why everybody's looking for other cheaper drugs that might block IL-1 as well as the IL-1 antagonist, but at least it is very safe.
Yeah. So my next question is, NLRP3 inflammasome also contributes to some of these diseases that we've talked about, but the inhibitor that was originally thought of to be able to be used displayed non-selective effects on cellular calcium homeostasis. So how did you then use this inhibitor as a chemical scaffold to build a series of inhibitors called the NBC series with the characteristic oxyborine ring, which then inhibit the NLRP3 inflammasome without affecting the calcium homeostasis?
Okay, so this was the work of my ex-PhD student, David Brough, who's now a professor. I've had over 50 PhD students, so although I sort of had a little input to it, it's really his work. So NLRP3 inflammasome is really important because the inflammasome is a scaffold of proteins that together are critical in driving inflammation. And it's the way that interleukin-1 is made. So previously we were targeting its actions. Targeting the inflammasomes is to try and stop it being produced. Now, the first inhibitors, as you say, they were quite non-specific. They had quite a lot of different actions. So what you do when you have a chemical that you know has a good property, but also has a bad property, you get clever chemists and clever drug designers, and they gradually modify the molecule, often one atom at a time or one group of atoms at a time, and usually test it in a screen in test tubes. So you need a readout. So your readout in this case would be stopping the production of IL-1. And perhaps your negative readback would be calcium changes. And you would maybe synthesize 100 molecules, each with slightly different changes. And each time you get a slightly better one of blocking IL-1 and less of calcium, then you modify it again and you do it again. Now, this is all done automatically, but it is quite a long process. And eventually you get to something that is much more specific for targeting one pathway and less so at the other. And this is important generically because in drug development, nearly all of the drugs we have are synthetic. Some are naturally occurring. You know, we get them from plants and things like that originally. And interleukin-1 antagonist is a naturally occurring. But most of them we synthesize. And so drug discovery is very much about tweaking those molecules and trying to get the one that's the most specific for that target and not having other effects. And then there are other properties you want, like you may want it to be very soluble, so you can just take it as a tablet, and you may want it to be absorbed, or you may want it to be particularly absorbed into fatty tissue, because you may want it to get into different tissues. And then you want it to have a very high affinity. In other words, a low amount of drug is needed to have a high amount of target. And so what Dave Bruff and his colleagues did with the chemists was gradually design new molecules that got more and more specific. Trouble is, when it's a new molecule, if you want to take that into humans, unlike a naturally occurring one, you've then got to go through all the studies of, is it safe? Does it have any unexpected toxic effects? But actually, they did find a drug that looked to be particularly effective, and it's a drug already in use. It's sold over the counter in some countries, called the Phenamate, and therefore it's already safe. So he's been working on that to see if that could be used as a treatment for Alzheimer's and other inflammatory conditions.
That would be brilliant. So my next question, we've already discussed this already a little bit, but IL-1, as we know, is a key contributor to the neuroinflammation and brain injury, yet the mechanisms by which it triggers this neural injury remain unknown. So to look at this, you induced conditional deletions of the IL-1R1 in brain endothelial cells, neurons, and blood cells in mice. So what did you find, and what significance does this have?
So what this was doing was trying to see, where's the target for IL-1? Is it the nerve cells in the brain? Is it the lining of the blood vessels, the endothelial cells, or is it other tissues? And with lots of people use now gene deletion, take your gene out and see what happens. But this is conditional deletion, means you can say you can stop having any IL-1 receptors on the blood vessels, but everywhere else, on the brain, none in the brain, everywhere else. So you very, very much targeted it. And what we found particularly was that it was the blood vessel cells that were important, as though IL-1 was acting particularly on those. Now, we know there are receptors for IL-1 on almost every cell, and sometimes taking them all out, you don't see any effect, perhaps because it's bad on one cell, good on another cell. A bit like the drugs that's non-specific. IL-1 has lots of different actions, and that's probably why taking all the receptors out didn't have a particularly beneficial effect. Okay. Whereas if you remove IL-1 itself, the gene, it does have a beneficial effect. And it looked as though it was particularly the blood vessels. And what we know is that IL-1 acts on cells in the brain, acts on neurons, but it doesn't kill them directly. As I said, it acts on the glial cells in the brain to release toxins which kill neurons, but it also acts on the cells lining the blood vessels. And there it has a number of different actions. It might release toxins. It probably damages those cells and makes them leaky, which is damaging, but it also causes the production of another group of molecules called adhesion molecules. And they're produced by the lining of the blood vessels, and adhesion molecules attract immune cells. So, for example, if you've got an inflamed cut, then the blood vessels in that inflamed cut will produce adhesion molecules that will attract the immune cells. They'll move into the tissue and they'll start to repair it because it's useful. Now, if that goes on too long, you get a chronic inflammation because you don't want them for long, and you actually probably don't want them going into the brain. But we know that circulating immune cells produced mainly in the bone marrow are very important in brain diseases, and they travel to the brain, they stick to the brain cells, the lining of the blood vessels, and then they can move into the brain. So that work suggested that the endothelial cells, the blood vessel cells, were particularly important as a target for IL-1. It wasn't conclusive, but it's likely. And the thing is, in biology, because I often compare it to physics. In physics, if you get an answer unless your equipment's wrong or you've done the experiment completely wrongly, you can be quite certain of it. In biology, you can do everything right and the answer might still not be right, because there's no two organisms that are the same. And that's why you have to do larger studies, you have to do them on different species. And in humans, it is the most complicated because you've got a huge mixture of genetic and environmental factors, age factors, gender factors, etc. And so anything in biology that's discovered, you need to say, well, was it just that situation it worked in, or is it more widely applicable? Can it be reproduced by others? Do you find the same thing in a different set of circumstances? So that means that is still quite preliminary, but it was still a big step forwards.
Mm. And the most recent inhibitor of this interleukin 1 is small thin graphene oxide. So can you tell us a bit about how this inhibits it and what the potential applications in biomedicine would be from it?
Okay, so again, this was Dave Bruff's work. So I should say first, what is graphene oxide? You might have heard of graphene, isolated in Manchester and won the Nobel Prize for 2 of our scientists. And it's a single atom thick layer of carbon. And it has remarkable properties. It's the strongest, most conductive of electricity, most flexible. It's remarkable. And actually, graphene oxide has some of the properties. Now, we don't actually know how it works in these experiments. We've got some ideas, but graphene is now being applied to medicine in a number of different ways. One way is tiny nanoparticles of graphene might be able to carry drugs to sites where it's active. And then graphene oxide, we think, also does have some anti-inflammatory properties itself, possibly direct physical interference. Not sure exactly why, but then there are much wider applications of graphene because it's so strong and so light. For example, replacement of joints. Rather than having a plastic knee joint or a hip joint, you might get a graphene or a graphene enhanced one. If somebody has to have brain surgery or has a major trauma where they normally have a titanium plate put on the head, and that can cause a lot of problems. Looking now at putting graphene plates, using graphene for electrodes, which are much more sensitive than the electrodes that we have. So there's a whole new field developed around the biomedical applications of graphene, and this was one of them. But exactly how it works, we don't know.
That's fascinating, and it's really wonderful and exciting for the possibilities that it might hold for the future. So do you have any sort of hopes for the near future in terms of what you might want to research next or look into? What are your sort of hopes for the future?
So, I mean, obviously, one of my hopes have been to, I am a biologist, and discovering new things has been important, but developing that as a new treatment, particularly for huge conditions. And one of the reasons I've been interested in inflammation, and we've got a big inflammation centre at the University of Manchester, is because it's important in so many diseases. It's important in chronic lung disease, in heart disease, in stroke, in Alzheimer's, in obesity and diabetes. So I'm hoping that some of the things that we've taken forward will be developed as new treatments in the future. But I should say, although I'm still involved in the research and I still go to the lab meetings, and many of the staff who train with me are now staff at the University of Manchester, I don't spend that much time on research now because, of course, I have to run the University of Manchester, which is huge. So that's more than a full-time job. So I sort of give a bit of advice when it's asked for. I comment on the results. I read some of the papers, but I'm not as involved, anything like as involved in it as I have been in the past. And what I do now is oversee quite a lot of research in all different fields at the university, whether it's environmental sustainability or the next applications of graphene, building a graphene plane, for example, or medieval history, maths, business, anything. So I only spend a little bit of my time on research now, but I'm still stay very interested in it. I started studying biochemistry and I switched to physiology. And the reason I switched was I realized I was particularly interested in the wider relevance to disease. So I was very switched on by a lecture about biochemical pathways, And I didn't find that very interesting. And then the lecturer started to put in context in saying, this is the pathway that happens if you've been starving for a while. And this is what happened in prisoners of war. And then when they were given food, it overactivated, and that's why it made them so ill. And I suddenly realized the context of the whole body physiology was what really interested me.
Yeah.
So rather than just the biochemical pathway, so you might switch.
Yeah, definitely. And I suppose natural sciences will give me that ability to choose. And yeah.
What are you studying for A levels?
Biochemistry and maths.
Okay. Good. Oh, well, good luck. Have you got any other questions?
No, that's everything. So thank you so much. I really
done it just in time.
Yeah.
Excellent. Well, good luck.
Thank you everyone for listening as well. I'll stop the recording.